Tuesday, we were talking about lethal genes or lethal alleles of a gene. And if an allele in homozygous condition results in lethality, I showed you a couple of examples where if you take a heterozygous individual that got this mutant allele, process to another heterozygous individual with the mutant allele, you end up not with a ratio that goes 3 to 1, like Mendel's ratios would have been, but a ratio that goes 2 to 1. Now this is simply an observation. We are observing the fact that there are 2 yellow mice and 1 black mouse in this cross. And based on that ratio, we are hypothesizing that this class is missing, correct? So how do you prove that this class is missing? What would be the follow-up experiment that you would do to prove that this class is missing? What did Mendel do, for example, to prove the genotypes of the F2s? He basically did another cross, correct? And in this instance, if I were to give you this question, what would you do? You would do a test cross, correct? Now, what would you cross against? You're supposed to cross against a homozygous recessive. So what's the homozygous recessive in this cross? Is it this one or this one? Well, you can't get this one because it's lethal, so you're going to cross it to the wild type homozygous. All right? And we're going to look at that with respect to the curly gene in flies. So you take the progeny of the previous cross and you perform a test cross with the homozygous recessive parent. So if you have a wild type fly and that homozygous recessive parent is the wild type fly. So if we take So we've got 3 classes, correct? We've got a class that's plus plus, that's normal wings. We've got a class that's plus minus, and that should have curly wings, and that's the 2. And then this is the class that we're predicting is lethal and missing. The homozygous curly mutant, correct? So we're going to take each of these flies. We're going to take the curly flies, and we're going to take the normal flies. Each one of these flies will be taken and then crossed to the homozygous recessive parent. In this instance, the homozygous recessive parent is basically the plus plus individual. So we're going to cross it to this individual, and we're going to ask the question in a test cross, what phenotypes do we observe? If you take this fly and cross it to the same genotype, plus plus to plus plus, all of the flies should be normal. If we take this class and cross it to this, 50% should be normal and 50% should be curly because it's a test cross of a single gene. And if this class is present, very important, if this class is present and you cross it to this class, you should get all curly, correct? But if we never find this class, then that would mean that this class is missing in that original cross, and that would argue that it wasn't lethal. So, you take the plus plus, cross it to the plus plus, you should get all normal flies. If you take the heterozygous curly, cross it to the homozygous recessive, 50% should be curly, 50% should be normal. And if you take the curly homozygous, if they are present and alive, and you were to cross it, you should get all curly. But this class is never found when you do this experiment. And because this class is never found, you can say with more certainty that this class is probably lethal in the cross, and it never produces live progeny. And so that's how you would be able to show that these crosses are completely lethal. So all curly flies from the previous cross will give you this ratio here, indicating they are all heterozygous curly. There are no homozygous curly coming out of this cross. And that will then argue that the homozygous curly is a lethal mutation, basically. So the 2-to-1 ratio is a suggestion, and then you have to do a second cross to prove that that is indeed the case, basically. All right. So homozygous lethal mutations arise in many different genes. For example, in yeast, there are 6,000 genes in the yeast genome, and of those 6,000 genes, about 2,000 genes are essential, i.e. they are lethal if you have a homozygous condition. So 1/3 of all the genes in a yeast cell are lethal. A very significant number of genes in human cells are lethal if they are mutated for both copies, basically, because these are essential genes that are required for the growth and development and the division of every cell in your body, basically. Things involved in replication, things involved in transcription, things involved in repair. If you mutate all of the copies, both copies, the cell dies, basically, and you don't get homozygous project. It's a very important class of mutants. All right? And most recessive mutants, unlike curly, do not have an additional dominant phenotype. They just have one single phenotype. So studying them is a little bit tricky. So what do we do to study them? We use a different class of mechanism for studying them. I'll show you that later on. Let's skip all this. We're going to skip this. This is just a sidebar. Fun sidebar that we don't have to worry about. Okay, so we're now going to talk about single gene. And I told you we're looking at diploid organisms, and that is correct. We are going to be looking at diploid organisms. So how many copies of the gene is present in a diploid organism? 2, correct? And so usually, not usually, always in a diploid organism, you have 2 alleles of a gene. They can be the same copies and the same allele, big A, or they can be different versions, big A, little a, correct? So, but you always have only 2 alleles in a single individual. But in a group of, say, 300 students, that gene, every one of you might have the same gene, gene A. But it doesn't mean all of you will only have either the big A or the little a or big A, little a, correct? You could have multiple forms of that one gene. So in a population, one gene can have multiple alleles. In an individual, one gene can have 2 forms. In a population, one gene can have, in theory, hundreds of forms, if not more than hundreds of forms. Every amino acid, in theory, can be changed into another 19 different amino acids. If a gene is 100 amino acids long, you can see how many different versions of the gene a single individual can have. Correct? So there is a humongous potential for different alleles to be present in a population of individuals. As I said, in a single individual, there are only 2 alleles, but in a population of individuals, you can have multiple alleles of a single gene. All right? And so we're going to look at multiple alleles and how they affect phenotypes as well. So a gene exists as one of 2 different states, or 2 different alleles, normal and mutant. And we define which one is normal and which one is mutant based on the prevalence in a population you're studying. So each of these states is, of course, the allele of a single gene. So red eyes, white eyes. One different allele, a second different allele. Smooth peas, wrinkled peas. One allele, another allele. But it is possible, and it is very common, for a gene to have more than 2 different forms in a population of individuals. Okay? Many genes will exist in 3 or more forms. And so when we see this, what we say is there are multiple alleles of a gene. All right. So it is important to remember, and I'm going to stress this over and over and over again, that even though a given gene has many different forms, in any single individual, you only have 2 copies. You're a diploid. So when you are investigating multiple alleles, you can only investigate them in pairs at a time. So if you have 3 alleles, you can investigate 2 at a time, then the next 2, then the next 2, and be able to understand how they all function. So here is our classic example proof. Okay, we're going to look at the white gene again in Drosophila. Okay? So here's the white gene, here's the wild type red gene in flies. Okay? The white gene is actually a protein that transports pigment into the facets of the eye. Okay? So the diagram is there is a pathway that makes brown pigment, there is a pathway that makes vermilion pigment. Both of those pigments are then transported into the facets of the eye via this transporter called the white gene. So when this transporter is working completely normally, you end up with red eyes. If this transporter doesn't work at all, you end up with white eyes. But if a transporter works kind of partially, it might transport one pigment but not the other, or it might transport one pigment to a greater extent than the other pigment, you start to get different colors in the eye. Okay? So how do we study this? Okay, many alleles exist for the white gene. The normal allele gives you red eyes. The completely mutant allele, i.e., let's imagine a situation where this gene is never made into a protein, the transporter isn't made, you end up with white eyes. But then you can also get intermediate alleles of that. One that gives you apricot colored eyes, and this is known as an apricot allele of the white gene, and it's written as little w with the suffix A. And similarly, you have other phenotypes. And right now, there are over 150 alleles of the white gene that we have investigated and analyzed. So you can imagine how many there are. And it's all to do with being able to detect changes in color in the eye. Okay?
Question?
Yes?
Is the transporter an enzyme or is it?
It's a protein. It's a protein. Okay? So it's in effect, it's like an enzyme, but it's not quite an enzyme. It just transports molecules from one cell to another, basically. So, yeah, it's got a function. In any individual fly, there are only 2 alleles. You can have big W, which is red color, and apricot together. Or you could have the little white and apricot together. Or you could have the big W and the little w together, and so on. Okay? So in any individual fly, you have 2 alleles, but in, say, 100 different flies, you can have multiple alleles. Okay, so how many genotypes are possible when you have 3 alleles at the white gene? Well, with 3 alleles, in pairwise combinations, you have basically 6 different possible combinations. You can have big W, that would be a red colored eye. You would have little w, that's a white colored eye. You could have big W, that would be red colored eye. You could have little WA, that's an apricot colored eye. You'd get a big W with a little WA. This would also be a red colored flower. And then you would have a little W, and that would be a, who knows, right? I'm going to leave that blank. I know the answer, but I'm going to leave it blank, because we're going to find out how you figure that out. So, we're going to look at the same kind of thing with rabbits. Okay? And there's a gene in rabbits for coat color, called the C gene. It's involved in the production of melanin. So, you have a full color when the gene is present in its normal form, big C. And that's this little rabbit here. Okay? Rabbits in the wild are not white. Okay? That's only your pet rabbits that are white. Rabbits in the wild, if they were white, they would get eaten up very quickly. They would kind of stand out, chomp, chomp. So, then there you have what's known as chinchilla, which is this one here, which is light gray in color. Then you have Himalayan, and that's this one here. It's an albino rabbit with black extremities. Its ears, its mouth, nozzle, and its paws are white. And then, of course, you have the one that everybody likes and loves, the little albino rabbit, which is a little C. So let's imagine there are 4 alleles for this single gene. Okay? 1, 2, 3, 4. All right. So how do we analyze these 4 alleles to figure out which one is dominant and which one is recessive? Well, as I said, you can only have 2 alleles in a single rabbit. So you make pairwise combinations. And then you look to see, in those pairwise combinations, what is the phenotype. Homozygous big C gives you full color. Homozygous chinchilla gives you light gray. Homozygous Himalayan gives you this phenotype. And homozygous albino gives you this phenotype. So we know what the homozygous phenotype is. But to figure out if an allele is dominant or recessive with respect to another allele, you have to combine the 2 in a heterozygous condition, correct? And so that's what you do. Basically take homozygous normal, cross it to a homozygous chinchilla. You get a heterozygous rabbit, and you ask what is the color of that heterozygous individual. Okay? So there are different alleles of the C locus, and we want to find out which one's dominant and which one's recessive with respect to each other. And what you will find when we do this experiment, and you'll see in a minute, is that there is a gradation of dominance to recessive across these alleles. And that gradation goes as full color is the most dominant, followed by chinchilla, followed by Himalayan, and then finally the most recessive is the albino. And the way you do this, you test the 2 alleles one at a time in a diploid. So big C is full color. Big C and chinchilla, full color. Big C and Himalayan, full color. I just charged these batteries just before coming to this. All right. So that's Himalayan, and then big C, little c is also full color. Then you do the next experiment. You do chinchilla with chinchilla, and that's of course chinchilla because that's homozygous. You do chinchilla with Himalayan, you make a diploid, and that's going to be chinchilla as well. If you have a heterozygous rabbit that's chinchilla with the little c, it's going to be chinchilla as well. If you then do Himalayan with Himalayan, it's Himalayan. And if you make a rabbit that's heterozygous Himalayan and little c, it will be Himalayan as well. And the only rabbit that's going to be albino is going to be little c. And so based on this, you can lay out the relationship. And you can say that the big C is the most dominant allele, followed by chinchilla, followed by Himalayan, followed by albino. And so you have a relationship of all the different alleles. Okay? So the C enzyme, it's an enzyme that makes tyrosinase. It's a tyrosinase that's the first step in the production of melanin. Okay? The black pigment, basically. And it breaks down tyrosine and helps produce the pigment that is involved in producing the normal colored gene. The chinchilla allele, the little CH allele, has only 20% of the activity of normal. So remember, normally enzymes have maximum activity and you have an excess of them. But when you're down to 20% activity, you produce less of the pigment. And so you get a light brown colored chinchilla rabbit. Now the Himalayan rabbit, mutant allele, is interesting because this allele is sensitive to temperature. It's able to function at one temperature, but not able to function at another temperature. And so it's active at low temperatures, and therefore the extremities, which are low temperatures, are the enzyme is active in those cells, and you get dark color because it produces the pigment. But the body is at a higher temperature, the enzyme is no longer active at the higher temperature, and you basically end up with white. And so that's why you have this. And then albino, of course, is the one mutant where the enzyme is not made at all. And so if the enzyme is not made, it's completely absent. You're not going to make any pigment. And that's why it's the recessive, most recessive gene. Okay? Is that okay? Clear? Good. Okay, so that's where we shift to another class of important mutants, and those are the temperature-sensitive mutants. Just like the C gene that we were talking about in the Himalayan version, that is sensitive to temperature. At one temperature, the enzyme is active. At another temperature, the enzyme is inactive. There's a mutation in the enzyme. So enzymes are proteins, correct? And they have a very specific structure. If you have a mutation that destabilizes the structure at one temperature but not the other, you may cause it to denature. And when it denatures, the protein no longer functions as an enzyme. In the instance of the Himalayan, the enzyme is denatured at a high temperature, but it's normally folded at a low temperature, and therefore it's active. And these mutants are called temperature-sensitive mutants. They are sensitive to temperature. Now remember, I also told you that most genes, essential genes, are difficult to study. The way we study essential genes are we try to make mutants in those genes that are temperature-sensitive mutants. So essential genes, you usually look for alleles that are temperature sensitive. So they function at low temperature but not at high temperature, or they function at high temperature but not at low temperature. Once you get a mutant like that for an essential gene, you can study the function of that enzyme better. And so, very important class, temperature sensitive mutations that we use. So they exhibit a phenotype at one temperature, but they are normal at a totally different temperature. Now you can have an enzyme or a gene that is functional at a low temperature and inactive at a high temperature, or vice versa. It could be the other way around. Correct? So it could go both directions. It's not just that it always is functional at high temperature and inactive at low temperature. It doesn't have that. It can go in either direction.
Question?
Yes?
Is this helpful because you could see genotype as phenotype?
It's helpful because, remember, essential genes, you can't study them in the homozygous condition, but you can if you have a temperature-sensitive allele, because what you do is you grow the individual initially at a low, let's imagine the enzyme is inactive at high temperature, okay? So you grow the organism at a low temperature in the lab, consistently, so it grows up nicely, then you shift it to a temperature that's kind of high. And you ask what happens to that organism when you shift the enzyme to a higher temperature. And in that way, you're able to study the function of the enzyme, basically. So that's how you are able to study these essential genes. So in yeast, for example, which is what I study most of the time, we study genes involved in replication. And so these are all essential genes. And the way you study them is you grow yeast at 20 degrees, so the cells grow. And then you shift them to 30 or 37 degrees. And the enzyme starts to get inactive, and you ask what happens to the yeast as the set temperature increases. So you go from 23 to 27 to 30 to 34 to 37, and you ask at what step does the enzyme become inactive? And when the enzyme becomes inactive, what happens to the cell? Is it able to replicate? Does it get arrested? At what stage does it get arrested? So in that way, you're able to understand the function of these enzymes. Okay? So these are conditional mutants. So cold-sensitive mutants, these are proteins that are functional at high temperature, but inactive at low temperature. And then there are heat-sensitive mutants. These are functional at low temperature, but inactive at high temperature. And you can get mutants that are CS or TS, cold-sensitive or heat-sensitive, in the same protein. So PCNA is a protein that's involved in replication, and it interacts with another protein called RFC. You don't have to worry about this. There's a mutation that converts the lysine to a glutamic acid. K is lysine in the single letter amino acid code, E is glutamic acid. And when you have this mutation, pCNA is cold sensitive. There is another mutant that converts a cysteine to a threonine, and that makes pCNA a temperature sensitive mutant. So you can see how mutations affect different functions and different aspects. And then once you have this mutant, you can begin to understand how it functions. And people have done lots of different experiments and figured out how that happens. Okay. So another important mutant that you find is coat color genes. And as I showed you with rabbits and also with cats, you have a gene in cats that is active at one temperature but inactive at another temperature. And it's active at the cool temperature but it's inactive in the warm temperature. And so you get light colored fur and dark colored fur, just like the rabbit example. So you get effects like this. Okay. So if you were to study this coat color gene in cats, you again will have the same sort of series that you had with rabbits. So big C, you get a full colored cat. It's a dominant, all other alleles. CB is the Burmese cat. Okay? It causes a lightening of the color, and it's only slightly temperature sensitive. CS, the Siamese factor, it's much greater lightening, and it's very temperature sensitive. And then little c is the most recessive form, produces the albino cat. Okay? Complete absence of any pigment and white fur. Now, if you just take CB and CS and analyze what's going on, CB is incompletely dominant over CS, okay, in the heterozygous condition, giving you an intermediate phenotype between Burmese and Siamese. So there is your incomplete dominance phenotype that you will observe. Question?
Is the wild type one just the black one?
Yes, dark color. It's not black, but it's dark color. And so this intermediate incomplete dominant cat is basically called Tonkinese. I'm going to offend half the class probably. I'm not a cat lover. I love dogs. Sorry. I know. I apologize in advance. All right, so we are done with, oh no, we're not done. We have one more little section to do. Multiple alleles, continuing on multiple alleles. Red blood cells. Okay, so we're going to keep talking about, question? No. Okay. So we're going to keep talking about multiple alleles and how they relate. And here I'm going to take you into the realm of medicine because I think this is important with respect to medicine. And so I'm going to bring this up. You've already seen multiple alleles and how you investigate multiple alleles. And we've also seen how you study you study proteins on red blood cells. You use antibodies, correct? We talked about it with respect to the M and the N antigen. Now we're going to talk about an antigen on the surface of a red blood cell. That's very important, and that's the ABO blood group. Okay? The ABO blood group, discovered in the same era as Mendel was, if you remember. And there are 4 blood types in humans. And these are defined by the formation of clumps. Right? So, serum, which is the liquid part of blood, and then the red blood cells, which are the cellular part of cells, part of the cellular part. There are white blood cells, which we'll ignore. We're talking about red blood cells. And these red blood cells have little antigens on their surface, protein molecules, or modified protein molecules in this case. And they basically are what we're going to investigate. Okay, so blood group is determined by a combination of the antigens that are present on the surface of the blood. So, when you're blood group A, you've basically got a genotype that will generate an antigen on the red blood surface A. And I'm going to show that as little green triangles here. Okay. Antigen on red blood cell is green triangles. Those are the proteins on the surface. In the serum, interestingly, you have antibodies that react not with the antigens A, but with the antigen B. So, in the serum, you have B antibodies, but A antigen on the surface of the red blood cell. in an individual with the blood group A. Individuals with blood group B, you basically have the B antigen on the red blood cell, and in your serum, you have antibodies against the A antigen. If you are AB, you are codominant. You have both antigens A and B on the surface of your red blood cell. All right? Clear? That's like the M and the N antigen.